This is the first set of notes on the IB internal assessment, which is commonly abbreviated as the IA. The IA is going to be a lab where students will design, conduct, analyze, and discuss a research question that you come up with all on your own. It has to be a unique lab, and you will do this in your senior year. Your research question will have to be approved by the IB Biology Year 2 teacher before you can get started on your lab. This is to ensure that no other student is testing the same topic and to make sure that your research question is in-depth enough to be able to successfully complete an internal assessment on that idea. You are able to have other students act as what's called helping hands or extra hands. So that means they can help you on certain things as long as it does not contribute to the thought process. Only your ideas can contribute to anything that requires thought or design. Examples of how they can help you are if you needed help gathering supplies, like gathering all the glassware that's needed for your experiment, or setting it up. They can help you with things like that. If you wanted them to peer edit, they can do that for you. So they can look for grammatical errors, typos. They can even look for parts of the lab that they find confusing, and they can say that this is confusing. But what they cannot say is this is how you should fix it. You need to come up with that fix. They can say things like, I think you're missing controlled variables, but they cannot say, you are missing these controlled variables. Okay, so again, it has to be your own thought process to fix it. So I would recommend that you find a non-biased student, somebody that's not in IB Bio or somebody that's doing a IB Bio lab that has nothing to do with what your lab is. Give them your procedure and have them try and set it up. That way, if they can't set it up, if there's anything confusing in that section, they can say, okay, I'm confused here. And then you can fix it before it gets submitted to IB. And your IB Year 2 teacher will give you feedback one time. But that feedback will also be similar to this. Again, the teacher cannot tell you how to fix it. They can only tell you areas of weakness that you need to go back and relook at. Your IA idea can be an extension of a lab that we've already done before, or it can be a new topic. So that's going to be up to you. We did labs in BioH. We're going to do lots of labs in IB Year 1 this year, and potentially there may be a lab towards the beginning of Year 2 you could use. But again, it's going to come very early. The IA comes very early in IB Year 2, so you need to be thinking about IA ideas now. Every time we do a lab, think, can I use this lab we're doing in class as a springboard to spin it somehow and come up with something new, something unique that we're not actually doing, and use that to help come up with an IA topic. And the same thing for labs that you did in biology honors. So there were lots of labs in that class, so maybe you could take some of those ideas and come up with a new IA topic. The IA will be assessed in 4 areas: research design, data analysis, conclusion, and evaluation. Each of these are worth a maximum of 6 points for 24 points possible. This lab will be 20% of your overall IB score next year. The remaining 80% comes from the external assessments. Those are the paper tests that you sign up for and take in May of your senior year. And there are several paper tests in biology that contribute to that 80%. So again, those are the external assessments, but the lab itself is the internal assessment. In this set of notes, I will be going over the research design section. There are rules to kind of the guidelines of formatting your IA. So you do need to come back to this slide every time we do a practice IA in this class, and then before you write your actual IA next year. So maybe circle, highlight this slide. It's pretty important. The written body of the IA is limited to 3,000 words. So you should not go over 3,000 words, but not everything is included in this word count. Okay, so below are the things that are not included in the word count. A data table. Okay, so if it's inside of a table, then that means it does not get included in the word count. That does not mean you can put everything in your lab in a table. There will be no more tables after the data analysis section. So everything after that section, the conclusion, the eval, that's definitely all written body. So that will count in the word count. Qualitative observations, you'll put those in a table anyways, and they won't count in your word count. No titles or headers or labels, those don't count in the word count. Sample calculations of your math, none of that counts. Citations do not count. And then the title page info that we'll talk about below, none of that counts in the word count. All of the font inside the lab report should be size 12. That means you cannot shrink down the font inside the data tables for the headers to make it fit better. You can't do that. Everything has to be size 12. And you have to pick the font that's something readable. So it doesn't have to be a very specific font. You can decide, but don't pick something cursive. Most students are going to pick Arial or Times New Roman. Your lab report does have to be double spaced for all the written body. There are things that don't have to be double spaced. I'd probably add this into these notes. Titles don't have to be double spaced. Anything inside of a table does not have to be double spaced, but everything else does have to be double spaced. So transition explanations between lab report sections, that does have to be double spaced. Your works cited does have to be double spaced. Sample calculations, that does have to be double spaced. Okay, so again, make sure you're following these guidelines. It does need to have 1-inch margins, and nothing should go outside of those 1-inch margins. So your data tables cannot be wider than the 1-inch margins. I do often see that in IAs, but you cannot do that. Your science does have to be cited. The rule of thumb is kind of, if it's common knowledge, you don't need to cite it. But if it isn't, if it's something you've learned over the years, or that maybe everybody doesn't know, or especially something you had to look up, it does have to get cited. If you're going to cite like your notes from Honors Bio or IB Biology, then do not cite your actual notes or your teacher. Instead, you will cite your current textbook. So that's how you would use that. Citations can be in whatever format you want. You don't have to pick MLA or APA specifically. I would pick one or the other and stick with it. Like, be consistent throughout your lab report. And you have to have a bibliography page at the end. So how you cite, and you do have to cite in text, but how you do that can be different. You can use numbers and then footnotes. You can use the parenthetical citations. Just be consistent throughout your lab. But do make sure you cite in text, and then you have a bibliography page that will come at the very end. Remember that bibliography page does not count in your word count because those are citations. If you use a web-based publication, make sure you have the URL as part of the citation, plus anything else that's needed. And then the date of access must also be included. The moderator has to be able to easily look up your sources, and they potentially could look up your sources. So if you don't give the exact URL of the date that you accessed it, then that could be a problem. For each section of the IA, you'll need to clearly explain why you did what you did. However, you do need to be concise with this because there isn't going to be room in your 3,000 word limit to kind of be fluffy with your explanations. You need to try to get to the point so you can be as concise as possible. Really try not to repeat yourself. That's a waste of a word count. So if you've already explained something in one section, don't re-explain it later. If you're, stating all your data, find a way to summarize it so that you can maybe put it all in like one sentence or a couple sentences rather than copying and pasting the same sentence over and over and over again so you can state all of your results. That would be redundant, again, and a waste of your word count. So you have to find ways to be concise. It seems like 3,000 words is a lot, and it should be plenty, it should be enough, but you're going to need those 3,000 words. Like you will probably be close to the 3,000 word limit by the time you're done with your IA. Avoid unnecessary or inefficient lead-ins, right? So like being really fluffy with how you transition into the next section could cost you words. So again, just be concise, remove unnecessary words, and just try to be simple with your language if possible. There is a link below that will maybe help you in some of these areas. And I will give you feedback this year too, as we go through your labs when I know you're going to be over the limit and you're lacking in one of these areas or more. I'll try to guide you like, oh, you are repeating yourself. You've already said this, things like that. You do need a title page. The title page can be a standalone page at the beginning, like page 0 of your lab report, or it can be at the top of your first page. It doesn't have to be its own page necessarily, but it does have to be at the beginning. The things that need to be included in your title page is a title of the investigation. This is not the same thing as the research question. Your research question is going to be very long and include a lot of important info, but your title is going to be a more simplified version of that, like a small statement of what your lab is going to be about. You won't put your name on your IA. Instead, you'll put your IB candidate code. I know you probably don't have this code yet, so in this class, when you don't have this code, we'll just use your student ID number. That way you practice using a number here instead of your name. And then your word count goes on the title page. The best way to get an accurate word count, since so much of your lab is not included in the word count, is when you're done with your IA, make a copy of it. On the copy, delete everything that I told you was not in the word count. Delete citations, delete titles, delete the data tables, delete your works cited. Okay, delete everything, sample calcs that's not included, and then count the words and put it on your title page. Okay, let's get into research design. Remember, you're going to get a score from 0 to 6, but in order to get to this score, you will be assessed in 3 different areas, which I will go through in a lot more detail throughout the rest of the presentation. We call these aspects. So there are 3 aspects you're going to be assessed in. The first aspect is the research question, but it's not just the research question alone. They're also going to be looking for, did you establish it with appropriate context? Like, does it make sense for the scientific context of your lab? Are you putting it into clear context? Does your research question make sense? Okay, so we are going to go through how you can make sure you're doing that. The second one is methodological considerations. And so this is not your actual methods, not your actual procedure. This is, did you consider everything that is part of the procedure to make sure your procedure will work and to make sure you can answer the research question with the data you're actually collecting? So are you going to be able to answer the research question? And how well did you do to make sure you are considering all aspects associated with that? And then the last, also safety and ethics will go into that aspect as well. The last aspect is going to be your actual procedure, your actual method. So is your methods repeatable, and how effective is your actual design? So let's kind of look at it from a rubric perspective. This is the IB rubric. So you're only looking here at the research design section of the rubric. There is a place here for you to put your total score out of 6 that you got for this section. Remember, there's going to be 4 sections on the lab report. This is one of them. Here's the 3 aspects I just talked about, and then descriptions of each aspect is underneath of them. You can get a score from 0 to 6 in any of the aspects. And then after you get 3 different scores here, so you're going to get a score for aspect 1, a score for aspect 2, and a score for aspect 3. After you get those 3 scores, the moderator is going to look at all 3 scores and decide overall what your score should be. So notice that there is like the score of a 1 or a 2 for the research question context has the same descriptor. So that does mean that there's some level of subjectivity, right? So if the research question is stated without context, then the moderator has to decide, are you on the low side of this or are you on the higher side of this? Like, it's not quite a 3, but it's better than a 1. It's not the weakest you could be in this box. And so they do have to decide between a 1 or a 2. Again, the same thing for the 3 or a 4. The research question is outlined within a broad context. So you could get a 3 or a 4 if you're being very broad with your research question, but the moderator has to decide, is it a 3 or a 4? Okay, like, did you barely make this category or are you almost to the next category? If it's described like well within the context, you could get a 5 or a 6. To get a 6 means it's perfect, right? Everything that went with it is perfect. It is challenging to get a 6. It's possible to get a 6 on some aspects for sure throughout your IA, especially the more practice we do, you'll get more confident and start to do better. But to get a 6 overall right here is definitely really hard because you would have to have a 6 in everything to get that 6. So again, we're going to do practice labs this year, and your job is to make sure you're reading my feedback and like striving to make those adjustments for your next practice IA, just trying to do a little better each time. So let's say I scored them a 3 for the research question context, so a 3 here. And then let's say I scored them a 4 for the methodological considerations, and then a 3 for their actual methods. So then we have 3, 4, 3. I then have to decide, what am I going to score them overall? So again, it's subjective, right? So I need to decide, is it more like a 3 or more like a 4? Since it's 2 3s and 1 4, it's going to be a 3. But even if it was 2 4s and 1 3, that doesn't make it a 4. So you really have to kind of look at overall this whole section, how is it going to be? A lot of times I err on the lower side simply because if you didn't hit that mark, you didn't hit that mark, right? If you didn't hit the 4 across the board, then you probably don't have a 4. When I put my score in the total box on the top right, I need to feel like IB would support that score. So sometimes I will round it up if I feel like it's really stronger than other labs that I've graded that had similar scores, but sometimes I won't, right? So you have to trust in the process that your teachers and the moderators have been trained to know the difference, to be able to tell what is a 3 overall for research design versus what is a 4. And that's how we score these labs. So again, I maybe suggest that you err on, the caution of rounding down when you're doing scoring for practice labs, unless you just think, well, it's really, really good, then maybe round up. Okay, let's get into all the details of each aspect. So aspect 1 is the research question context. So background information is going to go into this section, and it is important that your background information helps to put your research question and your methods into context. You essentially are going to be justifying and explaining scientifically what you are doing in the lab. How does your exploration relate to biology? And it needs to, right? You're not doing a chemistry IA, you're doing a biology IA. So those connections are definitely important. In the scientific background information, make sure you're telling the moderator any information they would need to understand like why you're doing your lab, why it's a biology lab, and to just understand the biology behind your lab. Okay, you can't assume that the moderator knows anything about your topic. But in this section, you need to limit the science to just what do they need to know to understand your lab. Make sure whatever you do explain, you explain it well. And don't put in a whole bunch of extra details that are not necessary. Remember, you need to be concise to save your word count because the background information counts in the word count. and then make sure you do have citations. So remember in Honors Bio, we did a lab with dialysis tubing. This was like a porous roll of dialysis tape, and we cut it into strips. We tied off one side and we opened it. So it was kind of like an open bag. Then we filled it with solutions and we tied off the other end of the bag. And essentially when we did this, we made a mock cell, a fake cell. And then we put them into different solutions. We did a diffusion lab like this, and we did an osmosis lab like this. So we actually did 2 different labs using dialysis tubing. So maybe you might think of something with dialysis tubing that you could do, because we definitely have those supplies here at Newbury Park High School. But this is the background information that somebody wrote for a dialysis tubing lab. Osmosis is the movement of water from hypertonic solutions to hypotonic solutions, a higher water potential to a lower water potential. And it is an important process in all living things. Without it, water would not enter root cells from the soil, nor would water be able to enter body cells. Dialysis tubing can be used as a model for a cell membrane. And then it says it continues on. So my question is, and don't forget citations, but my question is, if this was all they put for their background information, is that enough? And the answer is no, okay, because what they did not do is connect to their lab, right? So they wrote some scientific background information that's necessary to understand their lab, but they never actually connected to their design. So you must support your design. You need to justify everything that you're doing in your design. Controlled variables should be explained here. Why, right, are you controlling these variables? So here's some things that you would want to consider. What research helped you choose the range of independent variables? Because, for example, in the dialysis tubing lab, we did different concentrations of sucrose. For the osmosis lab, we did 0, 0.2 molar, 0.4 molar, and all the way up to 1.0 molar. So you would have to explain, if you were using similar concentrations, why did you pick those concentrations? Okay, because there's other concentrations you could have picked, but you specifically picked those, so explain why. Why did you control those particular variables? If you're saying you controlled temperature, why did you control temperature? Temperature affects the rate of osmosis, right? Warmer temperatures is going to speed up molecular motion. Colder temperatures is going to slow it down. So it's the why that has to get explained here. Why did you choose that many trials? Did you do 5 trials, 10 trials, whatever you picked? Explain why you chose that. How did you determine how long to run the experiment? Like, we ran our experiments for 30 minutes, but maybe you're running yours for a longer length of time. But there has to be a reason why you picked that. Explain that, justify that. Make sure you establish a connection between your independent variable and your dependent variable. That's very important. And then we're just trying to make sure we're supporting our fully focused research question. Keep in mind that your entire lab report is going to kind of flow. It should be a story that you're telling from beginning to end of your IA experience. And because of that, some of the aspects do kind of blend together and can get scored. It's not like specifically like, first you'll write up aspect 1, then you'll write up aspect 2, then you'll write up aspect 3. They do kind of mesh together. And so some of these explanations are going to get scored here, but some of them do kind of bleed over into aspect 2. It's important that you have a clear and focused research question. Your research question must include your independent variable plus all the levels and your dependent variable. What are you directly measuring? And so here's an example of an unfocused question. How do different concentrations affect osmosis? I know very little about what their experiment is about. Concentrations of what? Like, are they using salt concentrations, sucrose concentrations? I'm not sure. Right? Are they using dialysis tubing, like effective osmosis on what? I just don't know based on this particular research question. It's very unfocused. Here's a more focused research question. How do different concentrations of sucrose affect osmosis? So at least I know they're using sucrose, but this is still quite a bit unfocused. Here's one that's even more focused. How do the following sucrose solution concentrations, 0 molar, 0.2, 0.4, 0.6, 0.8, and 1.0, affect osmosis in a 1 centimeter cubed red delicious apple, genus and species are stated, that are left to soak for 24 hours? So this is quite a bit better. You can see that this experiment is not using dialysis tubing. They're using apple cubes. Okay, so we wouldn't have known that based on the previous unfocused questions. This research question is actually not fully focused. There are more things that need to go in this question, more details. The research question is long. How many trials are they doing? What is the independent variable? Like, it just says affect osmosis, but how are they measuring that? What else? The volume of solution. How much solution are they soaking in? Is that in a specific size beaker? There's just a lot that still needs to go into this. So if we were doing this experiment, then here are some of the things they would have answered in their background information. Why did you choose those concentrations, right? We kind of talked about that earlier. Why that type of apple? There's a lot of different species of apples, so why that one? This doesn't have to be something overly scientific. It might be, like you might have researched sucrose concentrations inside of different apples, since you know, whatever's inside the cell is going to affect osmosis. So if you did and you needed to use this specific apple, then explain that. But if that's not why, like if you picked it because this apple was easily accessible at your local grocery store or farmer's market, or because you have this apple tree, then, you know, it can be that too. But you do have to justify. Why 24 hours? I mean, we only let our experiment set for 30 minutes. So if you're choosing 24 hours, like you need to justify this length of time. Why sucrose? Why that size cube? And then, of course, what is the relationship between mass and osmosis? Because in this experiment, mass is going to have to be your dependent variable. IB doesn't specifically require a scientific hypothesis. Like, you can't lose points if you don't have one. However, it really helps your lab if you do have a hypothesis. So I strongly encourage you to have a scientific hypothesis. This is going to help you connect the independent variable to the dependent variable, which is required in the lab. You have to make that connection. So essentially, a hypothesis will be your answer to the research question. It should be explained, it should be testable, and it should be cited if you have an explanation. So here's an example. A worker on a fish farm notices that his trout seem to have more fish lice in the summer when the water levels are low, and he wants to find out why. His research leads him to believe that the amount of oxygen is the reason. Fish that are oxygen stressed tend to be more susceptible to disease and parasites. So here's the hypothesis. I think rainbow trout will suffer more lice in the summer during low water conditions because there is less oxygen in the water. Warmer water temperatures lead to a decrease in the solubility of oxygen. So not only are they saying what they think will happen and why they think that, but they're supporting it with science. So this is a well-written hypothesis. So it doesn't have to be a long paragraph, right? Be concise. Again, it's important that you do include a hypothesis because it makes a strong connection between your independent variable and dependent variable, which will help lead into your experimental design. This is not going to be the same thing as the mathematical hypotheses that we will set up in the data analysis section. Those are different. Those will be called the null and alternative hypotheses, so don't get those confused. We will need those to perform a statistical test on your data sets in the data analysis section. Aspect 2, methodological considerations. This is where your variables will get scored. So a variable is going to be anything that can change or exist in different amounts in your experiment. We're going to have 3 kinds of variables in our experiment: our independent variable, the dependent variable, and then controlled variables. The independent variable is going to be what you're changing in order to test or answer the research question, whereas the dependent variable will be the data that you're actually collecting. It's important that you report this as the data that you're actually collecting. Like whatever you're standing at the lab bench collecting immediately is your dependent variable. If you had to do math to get there, that's not the dependent variable. That means you processed the data. It's okay to state both as long as you clearly state the dependent variable. For example, if the dependent variable is the number of lice, you would need to state that, right? The number of fish lice. You would need to state that because that's what you directly counted. But you can say you will later use that to calculate the percent change in lice. Controlled variables are going to be everything else that you keep constant, because if you did not hold them constant, you wouldn't know what's causing the dependent variable to change. You wouldn't know if your independent variable is in fact causing that change. So we want to be able to answer our research question in confidence. Therefore, controlled variables are incredibly important. So same research question here. Our independent variable is going to be our levels of sucrose concentration. But when you state the independent variable, you need to state all the levels as well and the unit. That's really important. The dependent variable, again, you're going to state whatever you're directly collecting with a unit. So in this case, initial and final mass of the apple cubes, okay, and that would be in grams. Okay, so again, with a unit. And you can say, in order to determine the percent change in mass if you want. But you cannot just state that the dependent variable is the percent change in mass, because you didn't collect that directly. You had to do math to get that number. So it's important that you know what's correct and what's not correct in regards to that. For controlled variables, you want to list everything that would be considered a key controlled variable, meaning if you did not control it, it could affect the outcome of the experiment, but you don't want to list things that are not key controlled variables. So the type of apple, that's important, right? You can't use different species of apples if that's not your independent variable. Temperature affects the rate of osmosis, so you do need to control that. Volume, controlled. How long they soak, controlled. Equipment that takes the reading, so you don't have to use the same exact scale to mass it, but all the scales that you do use have to measure to the same level of precision. Like you can't use one scale that measures to 0.1 grams and then another scale that measures out to 0.01 grams. They must all measure out to the same level of precision. The method to prepare the cubes for massing is important. For example, if you are blotting them dry, then how you blot them dry, how many times you blot them, what are you using to blot them, same kind of paper towel that all absorb the same amount, all that's important. How you cut them, like what knife you use to cut them, like you wouldn't cut some with a serrated knife and others with a smooth knife because that changes the surface area, which could affect osmosis. Size and shape of the cube, some can't be pyramids and some are cubes. Make sure they're all the same, and how many cubes you put in each beaker. So those are all really important. Things that are not important to say, all independent variable levels are tested in the same room. That doesn't matter. If you needed a lot of space to test your experiment, you could use multiple classrooms. You just have to make sure you control the temperature, and you could use that using the thermostat. Humidity levels outside, well, our experiment is inside, so that doesn't matter. Same amount of light, doesn't matter. Light does not affect osmosis. The same person cutting the cubes doesn't matter as long as they're all doing it the same way. I recommend you put your controlled variables in a chart like this. It's like a data table, but it's not necessarily you don't have to call it a data table. Like we're not collecting any data, so you'll just call it a chart, controlled variables chart. So it doesn't need a lengthy title, and don't call it a data table, but you should label it as a controlled variables chart. And I do recommend you put it in a chart form because anything inside of a chart does not count as your word count, and so that would be good for you. Here shows you temperature, and then on the right side you have to say how you controlled it, not why. That's important because why was already explained and scored in aspect 1, right? You explained why in the background information. Here you're explaining how. So for temperature, why, you already said in the background information that higher temperatures increase the speed of osmosis. but here is how. So how did you control it? All the beakers containing the solution in apples were kept in the same room at 23 degrees Celsius. And always make sure you use the metric system. So many times students write why here. Please don't do that. You could put it in a list format too. That's you're not going to get scored down for this, but this would count in your word count. It's important that you make sure that the data that you're collecting for your lab report is both sufficient and relevant so that it can support your methods. Okay, so, and if you have to make solutions, you would have to show a sample calculation for one of them, like all those different concentrations that you made. How did you make a 0.2 molar solution? You would have to show one sample calculation and explain how you got that number, or how you got that amount of concentration, but you don't have to show it for all. You could just, explain how to substitute the other numbers to get the other concentrations. For sufficiency, that means two things. One, the different levels of independent variable that you chose to test have to be sufficient to answer the research question. So that means it's going to matter how many levels you choose, but that's going to be dependent on your actual research question. There's no set number. It's not like you have to have at least 3 or at least 5 levels of independent variable. You'll pick that. It will matter how you phrased your research question. For example, what is the optimal temperature? Well, optimal means at its best. That's a strong word. So if you pick that and you were doing temperature, you'd have a lot of levels of independent variable, and they'd all have to be very, very close increments together so that you could find the best temperature. I would avoid the word optimal if you can, because if you phrase the research question this way, what effect does temperature have on? That's really different, right? And you don't need as many levels of independent variable. So it's all in the phrasing. You do, however, have to have at least 5 trials of each level of independent variable. That's the minimum number, because this is going to allow for us to do statistical analysis. We're going to have to do standard deviation and then other statistical tests in the analysis section. And 5 trials at least of each level is required for that test to be valid. You won't have enough data otherwise. If you're running an IA on people, there's a lot of variability, so you would need a lot more trials. But you will have to decide what makes sense in regards to number of trials for your research question. But again, 5 is the minimum. So that means if we're doing 0 molar, 0.2, 0.4, 0.6, 0.8, and 1.0 concentrations of sucrose solution, that's 6 levels of independent variable. That's 6 times 5 trials each. That's 30 experiments that I'm running. So you need to consider that as well, like what's manageable and what makes sense for the research question, and can you actually get it done? The data also has to be relevant, meaning it actually can answer the research question. So an example of making sure your data is relevant is shown here. If you were going to measure the amount of bacteria that grows on the desks as the day goes on, you'd have to sanitize the desk first, right? And it would have to be the same for every desk you're testing, however many sprays you use on that desk, and the amount of sanitizer or the type of sanitizer that you're using. All of that has to be very much controlled. If you just start measuring without sanitizing first, then you can't really compare because you don't know how much bacteria is already there. So again, your data has to be relevant too. If you're conducting field work, like out in the field, like outside, right? Like you're going to a park to collect your data, you have to make sure your sample is random, and there are ways to do this. Like if you were counting, I don't know, dandelions at the park, you can't go find a patch of dandelions and walk straight to it. You have to have a random way to sample, that patch of land. And so when we do ecology, we will discuss ways to ensure random sampling. So I'll come back to that. Safety, ethics, and environmental issues have to get discussed here and how you handled them. You may not have some of these issues, in which case you will explain why you don't have some of these issues. You'll almost always have safety issues, like if you work with glassware or hot plates, that you can talk about. If you work with humans, like you're doing an exercise lab, then each human does have to have a signed permission slip. If you're working with animals, you cannot take them outside of their normal range. So for example, if you were like, I want to test different pH levels on goldfish. Well, IB has a very strict IB experimentation policy on animals. And if you're taking a goldfish out of its normal range of pH levels, it will die. And you would know it would die because that's outside of its natural range. You're not going to be able to do that lab. Your teacher will never approve a lab where you're taking an animal outside of its range because you're knowingly doing harm to that organism, and that is not allowed. So you do have to be very careful if you're working with animals. You do have to make sure you're always disposing of everything safely too. So proper disposal of chemicals to be safe, of organisms to be safe. All of that definitely matters. You may have environmental issues with your species. So you need to make sure that whatever species you're using is not invasive. And if it is invasive, which is okay, you can use invasive species. You just have more to consider when you dispose of them, right? Otherwise, you'd have an environmental issue. You don't want to release something into the wild that could take over because it's invasive. So if you're working with invasive plants, for example, maybe you'll freeze them until they die and then you can throw them away. Or maybe you will let them completely dry out until they turn brown and die, and then you can throw them away. But you're not just going to put them in a pond because they could take over. If you don't have any issues, remember, you need to explain why. For example, if you're doing an experiment with plants and some of your plants die, that's actually considered okay because plants don't have a nervous system, so they cannot feel any pain. So there's no ethical issue there. But with animals, there would be an ethical issue, right? That's why they have that policy in place. All right, aspect 3, the description of the methods. So this is where you're going to get scored on your actual procedure and anything that's directly related to the procedure. So one thing that you will need before the procedure is a list of all of your materials, and you need to be specific. Like don't say a random container, right? You need to say a 500 milliliter clear glass beaker. And in addition to that, if you need 5 of those, you should say 5 500 milliliter clear glass beakers. And a beaker is going to have equipment uncertainty. So that should be stated in this list as well. Whatever the uncertainty of that beaker is, you would state it. And that's not something we've practiced yet, but we will start practicing that in this class. So I will explain to you how you will be able to figure out equipment uncertainty for the different kinds of equipment that you're going to use in this class or next year. So make sure you're specific with that list. The moderator should be able to take your list, go gather all the supplies, have it all ready to go, and then they're ready to start your procedure, right? They'll already have everything that they need. They're not going to have to go back to a cabinet and grab something out later. Make sure it's complete. You need a diagram of your setup. This can be a picture that you have, labeled somehow, or it can be a sketch or drawing or some sort of diagram of your experimental setup. It does have to be clearly labeled, and it needs a title. So you can see figure 1, and then they're putting a title underneath of it because figure titles go below the figures. You also need to have step-by-step a numbered procedure, step 1, step 2, step 3. If you're going to say that you're doing 5 trials of a particular level of independent variable, you don't have to copy and paste all that over and over and over again. That'd be redundant. You can have a step that says repeat steps, 5 through 10 for trials 2 through 5, right? You can just say that. If you're doing the same thing with different levels of independent variable, then you can have another step that says, repeat steps this through this, but for 0.4 molar concentration, right, for 5 trials. So that way you're being concise because your procedure counts in your word count. If you do any preliminary trials, like you have a mock lab that you set up ahead of time just to see if your lab's even going to work, then you would need to discuss that also. Not all students will have that, but if you've invested time into that, it should go into this section as well. When you write your step-by-step procedure, it's important your procedure is readable, very important, so that a competent scientist, or AKA the moderator, could in fact repeat your identical procedure without any questions or concerns. So try to be very thorough while still being concise at the same time. And that is the end of this section of notes.